I'm Danielle Kuhn, and this is my presentation on spinal muscular atrophy type 1. So what is SMA type 1? It is a type of neuromuscular disorder, and its mode of inheritance is autosomal recessive. So this means that this disorder tends to skip generations, and two copies of the gene must be present to have this recessive trait. And the most common form of this is a homozygous deletion of the SM1 exon 7. So a homozygous deletion would consist of multiple nucleotides within the gene being extracted from the gene. Because this is a homozygous deletion, both copies of the gene are no longer present because it is homozygous. And as you can see in the figure I provided, the SMN2 is untouched in this process, and the SM1 gene is absent here in this gene deletion versus the mutant would consist of SMN1 not being affected by the deletion. And so with the homozygous deletion of the SNN1 exon 7 consisting of an exon, exons code for different proteins after alternative splicing. And alternative splicing is when a single pre-mRNA is processed in various ways by removing the introns and splicing together the exons in various ways to produce different proteins. So for this mutant, SMN protein is vital for the alpha motor neurons to function, and this allows for the muscles to grow and move and all those things. With the deletion in this SN1 gene, no SMN protein is made. Therefore, the alpha motor neurons cannot function. So this is kind of how it works in the gram scene of it on a molecular level to how it actually presents. So we have the autosomal recessive inheritance, requires those two copies for it to be present. And then we have the homozygous deletion of the SNM1 gene, exon 7, that therefore loses creates a loss in function of the SMN proteins that are required for the alpha motor neurons to successfully do their job of moving us and creating muscle and allowing us to have those abilities. But unfortunately, because of the deletion of the SNM1 gene, we lose the function in the SMN protein, and then there's a degeneration of those alpha motor neurons, which then leads to SMA type 1. So in a wild type, we would have both copies of the SNM1 gene. Instead, in our mutant, our copies are deleted, both of them. And as you can see in the figure, this is just a wild type. It just shows how we need the functional SMN protein in this process that SMN1 creates. So in our mutant, our protein is no longer able to be made. Therefore, we result in the degeneration of alpha motor neurons, and SMN1A type 1 is developed, and here comes an array of symptoms. So as you can see in the photo, there's a lack of head control and reflexes, and there's a low muscle tone. And this is because we do not have those motor neurons activated to the best of their ability. And with that, a lot of the key features in this disease, in this disorder, is that it affects a lot of our vital muscles that are needed for things like breathing and eating. So patients that have these have trouble with these daily processes, and they have unstable sitting. As we can see, the baby is slouching down because it lacks those proper muscle tones. And its prevalence in America is that 1 in 10,000 births have a form of SM in the United States altogether, because there's different types of them. However, of those cases, 45% of them are type 1. So there is about the most common type of SMA within the United States. So as we can see it on a chromosome, it's in the long arm of chromosome 5. So here's a photo of chromosome 5. So it would be in the long arms of the chromosomes. And in this figure I've also provided, it is within the FQ13.2 region. And here's chromosome 5 with that region. And as we can see, both the SMN1 gene in blue and SMN12 gene are present on this chromosome in this region. And in this mutant, we would not have the SMN1 gene. And PCR amplification is a vital process in order to identify these mutations in the SMN1 gene that I've had the great ability to conduct in lab. So PCR is a polymerase chain reaction and is a technique used to amplify, which means to make several copies of a DNA sequence. And in the context of SMN1, long-range PCR amplification for SMN1 is used, and then DNA sequencing is used after that in order to detect these mutations for people to be aware of this condition that they might have or might have one in their family, so that they can plan properly. And the treatments for this disorder involves physical and occupational therapy, getting those assisted services in order to properly be able to function in everyday life. Ventilation devices, as I said before, a lot of the muscles that are vital for our survival, such as breathing, we need help combating those. And also feeding tubes, same goes with that. Being able to do that daily function in life and being able to get these people accommodated to this disorder is very important for this. Here are my citations. Thank you for listening.